Hello friends, myself Hitesh Shahare, working as an assistant professor at SNJB's Shriman Surya Jain College of Pharmacy, Chandwad, Nashik. I welcome you all. In this session, we are going to discuss about the antibiotics and in detail about the penicillin. So, antibiotics, the first time this term was renowned by scientist Waxman for the discovery of streptomycin. This antibiotic was used in 1952 for anti-tubercular action and the scientist got Nobel Prize for this discovery. So antibiotics can be defined as any substance which is produced or metabolically derived by the fermentation of living organism and it is capable of inhibiting the growth or survival of one or more species of microorganism in high dilution. Now how this antibiotic term was discovered? So microorganism when produce the substances and that substances act against other microorganism, that is known as antibiosis. So similarly from this antibiotic term is discovered. Now when we can start this antibiotic treatment? So when the bacteria multiply rapidly, like within 2 to 3 hours it become double, then there is need to initiate the antibiotic therapy with high dose, that means our loading dose, and that last with the maintenance dose. Now antibiotics are classified based on the various parameters such as source, spectrum of activity, mode of action, chemical nature and effect. So take a look one by one how these are classified. So based on sources, the antibiotics are classified as natural source, semi-synthetic and synthetic. Natural source means the antibiotic obtained in large scale by fermentation of microorganism. Example, bacitracin. They are totally from the naturally. Now semi-synthetic means we are having naturally obtained drug, then we are carrying the modification in that structure by synthetic way, so that becomes semi-synthetic. So example, ampicillin, when we carry out the modification in side chain which is present at the 6th position of amino penicillinic acid. Synthetic means which obtained totally from the synthetic source, synthetic method. Example, chloramphenicol. Another way of classification of antibiotic, that is mode of action. So drug which inhibit the biosynthesis of bacterial cell wall, that is one way of class. Second, drug which interfere with nucleic acid synthesis in bacteria. Third, as drug which inhibit the protein synthesis in bacteria. Next, drug which interfere with the cytoplasmic membrane of bacteria. And last, drug which interfere with the folic acid synthesis, that means folate antagonist. Take a look on the example of each. So these are all example of these different classes of antibiotic which are classified based on the mode of action. Spectrum of activity. These antibiotics are classified as narrow spectrum as well broad spectrum. What mean by narrow spectrum antibiotic? This antibiotic having the activity against only one species of microorganism, while broad spectrum means they act against both species of microorganism. One species of microorganism means they act either only gram positive or only gram negative. These are having high degree of selectivity toward the target because they are act against only one species. Example, bacitracin. Broad spectrum act for the gram positive as well gram negative microorganism, and because of this, these are having less degree of selectivity toward their target. Example, chloramphenicol. Again, the antibiotics are classified based on the effect of activity, what kind of effect they produce, either bacteriostatic or bactericidal. Now, what mean by bacteriostatic? Static means here the inhibit. So they inhibit the growth of bacteria, not kill. Example, sulfonamide and tetracycline and many more. While cidal means which kill the bacteria totally. Example, aminoglycoside, penicillin, cephalosporin and many more. Now, this is important in view of chemistry, the chemical nature, chemically classification of antibiotic. So first one here, the beta-lactam antibiotic, the major class of the antibiotic, beta-lactam antibiotic. It is again having subclasses: penicillin, cephalosporin, thienamycin, clavulanic acid, sulbactam, monobactam, azobactam and many more. Aminoglycosides, example streptomycin, kanamycin, tobramycin. Tetracycline analogues, tetracycline, oxytetracycline, minocycline, rolitetracycline. Next is macrolide, the example azithromycin, erythromycin. Next chemical class is polypeptide, which contains the peptide chain, long peptide chain, bacitracin. Next, pauline antibiotics, amphotericin B, nystatin. Next chemical class is lincomycin analogues, example lincomycin. And lastly, the unclassified antibiotic, in that chloramphenicol, rifampicin, linezolid. These are certain example. So this is about the introduction part of antibiotic, in which we have discussed about the what are the antibiotic and how they are classified. Now, based on the chemical nature, as we have seen beta-lactam antibiotics, so we are going to talk here about the penicillin antibiotics. So let us start with this first chemical class, that is the beta-lactam antibiotics. So in this we are going to discuss about the introduction of beta-lactam antibiotic and how these are acting, classification of beta-lactam antibiotic and their development, chemistry of penicillin, therapeutic uses and the adverse effect of penicillin. So beta-lactam antibiotics here contain a lactam ring, that means a cyclic amide. This is what a lactam ring, cyclic amide, NHCO. So beta-lactam ring fuse with other heterocyclic ring and gives various beta-lactam antibiotics. So here the second heterocycle is fused to the beta-lactam ring. Beta-lactam ring, when fused with the thiazolidine second heterocyclic ring, that gives penicillin kind of antibiotic. When beta-lactam ring fused with the second heterocycle ring, dihydrothiazine, that gives cephalosporin. The same beta-lactam ring when fused with the oxazolidine we are talking about this second heterocyclic ring here fused. Take an example here, dihydrothiazine, then it gives cephalosporin. Here if oxazolidine is there, then that gives us the clavulanic acid. Beta-lactam ring here when fused with the pyrroline here, that gives a thienamycin. So these are the major one. Many more are there. When we change here the second heterocyclic ring, we are getting the number of molecules. This beta-lactam ring portion is very important to bind with the proteins which are present in a bacteria, and that is responsible for action of beta-lactam antibiotics. So this is about the sub-classification of beta-lactam antibiotic. Mainly these are classified as penicillin, cephalosporin, carbapenem, monobactam, and beta-lactam is inhibitor. These are the certain example of each classes. Cephalosporin are classified again first generation, second generation, third generation, fourth generation. Example I have mentioned over here: carbapenem, monobactam, and beta-lactam is inhibitor. These are classified again here. These are example I are having clavulanic acid, sulbactam, tazobactam. So take a look on how these beta-lactam antibiotic act. So this is a common mode of action for all these kind of beta-lactam antibiotics. So as we know, bacterial cell wall is composed of peptidoglycan. This is a peptidoglycan layer which is made by two amino sugars. The one N-acetylmuramic acid and second is N-acetylglucosamine. These are the two sugars which form the peptidoglycan layer. This is NAM and NAG. So transpeptidase is one of the enzymes which is present in this bacterial cell that carry out the cross-linking of these two sugar and form amino acid chain. So here you can see the cross-linking of this by the enzyme of these two sugars and they form the amino acid chain, that means a peptidoglycan layer. That is responsible for formation of bacterial cell wall. When beta-lactam antibiotic bind to this enzyme, it not allow the cross-linking of amino acid to form the peptidoglycan layer. That means they inhibit the synthesis of bacterial cell wall and that result in death of bacteria. So likewise, penicillin or beta-lactam antibiotic act as a bactericidal. So this is all about the mode of action of all kind of beta-lactam antibiotics which produces the bactericidal action. So coming to the first, penicillin, as I said, we are going to discuss in detail about the penicillin over here. So penicillin obtained from the Penicillium notatum. So let us take a look on this. This is the general structure. What are the penicillin? They contain the beta-lactam ring. As I told you here, they belong to the beta-lactam class of antibiotics. So they contain beta-lactam ring. It is a cyclic amide. You can see here. So they are fused. This beta-lactam ring is fused with the second heterocycle, that is the thiazolidine, and that gives penicillin. So here, that's the general structure for this. So it is a beta-lactam ring. Second, it is a thiazolidine ring which is fused to the ring, cyclic amide ring. So this combination, this complex is known as penam. When there is a double bond over here, it will become a pename. So these penicillin are isolated from either Penicillium chrysogenum or Penicillium notatum, and it was discovered in 1928 by the scientist Alexander Fleming. So this was a serendipitous discovery. So let's take a look on discovery of penicillin. This is a one structure, common structure. So this is the scientist Alexander Fleming who has discovered the activity of penicillin. Later on, the two scientists Florey and Chen, who has carried out the clinical trial in 1940 on the same and got Nobel Prize in 1945 for its action. So Alexander Fleming has kept the petri plate of Staphylococcus aureus in his laboratory near to the window, and on next day when he came to the lab, he found some zone of inhibition in that culture of Staphylococcus aureus. He worked on that and found that the plate was surrounded with the some of mold. That mold was a Penicillin mold, and he thought that that compound may be isolated from Penicillin mold that inhibit certain culture of Staphylococcus aureus and shown the zone of inhibition. And then by this he has isolated the one compound, and there was a discovery of penicillin. During the Second World War, this penicillin saved many lives as they used for infected wounds. During that time, it was used to treat diphtheria, gangrene, pneumonia, syphilis, and tuberculosis. Now, how these antibiotics were developed? So these were developed in certain ways, like first there were the natural antibiotics which lack the oral bioavailability because of susceptibility to hydrolysis. When they used to take by oral route, easily susceptible for hydrolysis by the acidic condition as well basic condition, and because of certain enzyme. So penicillin G was the example of natural penicillin. Semi-synthetic, here second generation. Then why there was need to develop semi-synthetic antibiotic, semi-synthetic penicillin? Because penicillin has certain drawback such as poor oral efficacy, susceptibility to penicillinase enzyme for hydrolysis, not stable in gastric pH, rapidly broken down into the stomach when taken by oral route, narrow spectrum of activity, and hypersensitive reactions. So there was need to develop in a semi-synthetic molecule, modifications to be done in penicillin structure. So semi-synthetic are having with the enhanced oral bioavailability. They are resistant to the enzyme hydrolysis, example naphthalene. Again, these are developed as a third generation aminopenicillin. What mean by this here? So there is an introduction of amino group in a side chain, and that is responsible for enhancing the oral bioavailability than the previous two, greater efficacy, and they are resistant to the hydrolysis in acidic condition, basic condition, as well in a enzymatic condition, example amoxicillin. Again, these were developed by changing the amino group. There is the introduction of carboxy group in side chain rather than the amino group, and these are with again more enhanced oral bioavailability as well greater efficacy, and they are resistant to the hydrolysis, example carbenicillin. So this is a general structure for penicillin. This is one position, second position, fourth position, 7 here. Now there are 3 chiral carbons, carbon 3, carbon 5, and carbon 6. So these are 3 chiral carbon present in a general structure of penicillin. The carbon bearing amino group at the carbon 6, this is amino group which is present at the carbon 6, has L configuration. Carbon bearing carboxyl group here has D configuration. Acyl amino group and carboxyl group. So here there is a amino group and this is a carboxyl group are trans to each other. These all natural penicillins are dextrorotatory. If we see the nomenclature as per CAS, chemical abstract and USP, so chemical abstract, by that way, the sulfur is denoted as a number 1 position, while in case of USP, the nitrogen is denoted as a number 1 position. Now penicillins are classified as naturally obtained and semi-synthetic, means penicillin obtained from Penicillium chrysogenum or Penicillium notatum, that is the natural source. What the penicillin are having drawback, that need to modify, so there is need to develop a semi-synthetic molecule. So modifications were done in a side chain of penicillin molecule and obtained the semi-synthetic antibiotics. So natural obtained drug here, the one which are acid labile, means not resistant to the acidic condition, they are rapidly hydrolyzed in acidic condition, and then these were modified again. Certain are there which are obtained from natural source also, which are acid stable. So example for this, penicillin G and acid resistant as penicillin V. Now in case of semi-synthetic, so one as beta lactamase inhibitor, this is one of the enzyme lactamase which cause hydrolysis, which broken down the beta lactam ring. So that lactamase enzyme is inhibited, which is present in a bacteria. So example here, clavulanic acid, sulbactam. Penicillinase resistance, this is another enzyme which is present in bacteria and which also cause hydrolysis of the penicillin. Example, methicillin, cloxacillin, flucloxacillin. Extended spectrum, the antibiotic which are developed as having extended spectrum of activity. These are of in subtype aminopenicillin by the introduction of amino group in penicillin structure, ampicillin, amoxicillin, introduction of carboxy group instead of amino, that is a carbonicillin, and uridopenicillin, the example piperacillin. So this is about the classification of penicillin as in natural and semi-synthetic. Now this penicillin, how they degrade. So this is what the penicillin structure, 6 APA molecule. You can see here the 6th position containing the acyl amino side chain. So in basic condition as well, in presence of the enzyme, these are hydrolyzed into the penicilloic acid. You can see this bond is here broken down, this bond is here broken down. That beta lactam ring is broken down by the role of basic catalyst or basic conditions or the enzymatic condition. If we talk about the other one more, that is the acidic condition, in that this ring is totally broken, 2 rings are separated, and that gives a penicillamine as phenylaldehyde. These 2 molecules are obtained in acidic condition by hydrolysis. This penicillic acid on further heat and on decarboxylation, it gives a phenyloic acid. This phenyloic acid is again on reduction gives a 2 molecule penicillamine and phenylaldehyde, which we are obtained on hydrolysis of penicillin in presence of strong acid. So this is a degradation of penicillin. Now coming to the SAR, this is what a general structure. This is a beta lactam ring. The second heterocyclic ring which is fused to the beta lactam ring, that is the thiazolidine and gives us a penicillin. And third part as a acyl amino side chain. This is acyl amino. So these are the 3 important parts of the penicillin structure: beta lactam ring, thiazolidine and acyl amino side chain. So these all, thiazolidine ring, beta lactam ring and NHCO, this is a common part in all the structure of penicillin analogues. Just we need to modify this R and we are getting the number of molecules. So start with this. First position, sulfur atom at position 1 when oxidized to the sulfone SO2 or sulfoxide, that is stable to the acid condition, but it is with the reduced activity. So this is to be only maintained as a sulfur, not to convert in a sulfone or sulfoxide, because it gives less activity. Second position, it contains the dimethyl group here, CH3 CH3. These are essential for activity, and if any change is here, that reduces the activity. Third position, this is a chiral carbon which contains COH. So here carboxylic acid is again essential for activity and required penicillin binding protein recognition. This is very important for target binding. This carboxylate ion bind to the charged nitrogen of lysine residues which are present in a binding site. So if change to the ester, that become a prodrug and bioavailability increase. So we can make a change over here only to convert the ester. So again, coming to the next point, fourth point, as it is containing nitrogen. Now coming to the fifth position of this, so that should be unsubstituted. Coming to the sixth position, that is a chiral carbon here again. So that can be substituted with the variable group on amide side chain. That means we are talking about this R. This is amide side chain, CONH. Now here there is a R. That means a group. What it can be? What we can substitute here? So firstly we can say about the electron withdrawing group. We can substitute EWG, electron withdrawing group, that increases the stability toward the acid because the amide oxygen become less nucleophilic. Again, what we can substitute here? We can substitute the bulky group here in amide side chain that make more resistant to the beta lactamase enzyme. There is a steric hindrance which protect from the beta lactamase enzyme. Now here again we can substitute the polar ionized group. At sixth position we can substitute again polar group, ionized group or acidic group that make more active against gram negative bacteria. Now coming to the position 7 over here, that contain the carbonyl group. It forms the resonance stabilized structure for nucleophilic attack. So that is again essential. So this is about the 7 point. Now position 6, carbon 5 here and carbon 6 about this. So that should show cis kind of stereochemistry because it is essential for activity. So this is all about the SAR of penicillin. We have seen from 1 position to the 7 position. So this is a summary part what we have seen just now. So this is of site of enzyme action on the beta lactam ring, which is broken down this. Next, this carboxylate is very important for the activity. We can convert this into the ester that make a prodrug. This COH is important for the binding with the target. This dimethyl group here essential. This carbon 5 should be unsubstituted. This carbon 5 and carbon 6 having cis kind of stereochemistry. This acyl amino group here, R group, should be substituted with the polar group, ionized group, acidic group. We can substitute here the electron withdrawing group. So this is all about the SAR of penicillin. Now let us take a look on different example of penicillin analogues. So this is what first penicillin naturally obtained, penicillin G. It name as benzyl penicillin. So we should know benzyl structure. What the benzyl structure? Just take a look. This is a benzyl. That only we have to add here, and the rest of part is common in all kind of penicillin analogues. Just we have to change the R. So here the name only indicating benzyl means we have to substitute only benzyl, CH2 and phenyl ring. That only we have to add here, and we are getting benzyl penicillin. Next is phenoxymethyl penicillin. It is named as penicillin V. Phenoxymethyl. If we know the benzyl penicillin, it will be easy to get the next molecule, phenoxymethyl. Just what we have to introduce here, oxygen between the benzene ring We have to introduce here oxygen between the benzene ring and between the CH2 what here we have added. Okay, so this is what the phenoxymethyl. Phenoxy means phenyl ring with oxygen and methyl CH2. So if you know benzyl penicillin, it will be easy to get the phenoxymethyl. So by this way we can remember, if you want to get a series of compound, then you have to follow some sequence for the structure. So from one you can make the another analog. These are the marketed preparation. Coming to the amino penicillin means introduction of amino group. So here again you should know about the structure of benzyl penicillin. What benzyl penicillin is? CH2 and benzene ring as a R. So what the CH2 is present in benzyl penicillin, that one of the hydrogen of CH2 is replaced by the NH2. You can see here benzyl penicillin. So CH2 was there, now hydrogen is replaced with the introduction of NH2. So it become amino penicillin. So it is a ampicillin. It is a ampicillin AM indicating the presence of amino group. So here you can see next, if you know ampicillin, it will be easy to get the next molecule, that is amoxicillin. Amox, AM for indicating the presence of amino group. So you can see it is a amino penicillin. OX, that indicate the presence of hydroxy group. You can see here hydroxy. So if you know this ampicillin, it will be easy to get this amoxicillin. Just you have to introduce hydroxy group at para position of this ring and you are getting the amoxicillin. So by that way you can remember from one to another. So these are the marketed preparation. Next is carboxypenicillin, that again you can remember, just there is a replacement of amino group with the COH and you are getting carboxypenicillin. So if you know the ampicillin structure, it will be easy to get the structure for carbapenicillin. Carba indicating here presence of carboxy group. So take a look on this. This is what there was a NH2 here, now it is replaced with the COH. Rest of is same as ampicillin. So this is a carbapenicillin. Next is ticarcillin. Here what you have to replace this benzene ring, here you have to replace with the thiazole. Now how you can remember this thiophene. So here T, the T indicate the presence of thiophene, car indicate the presence of carboxy group. So here you are getting this ticarcillin. Rest of is same. These are the marketed preparation. Now if we talk about penicillinase, the enzyme resistant drug, methicillin. The word indicate methi, METH, that indicate the presence of methoxy group here. Now here you forget about the structure of benzyl penicillin. Coming to the directly NHCOR. NHCO, here NHCO and R. Okay, so here NHCOR, you can see here the R is now changed, not there is CH2 and phenyl ring. Now here NHCOR adds directly here phenyl ring and that is substituted with the OCSE, so that indicate the methicillin. Now here next naphthalene, Na, they indicate the presence of naphthalene ring. You can see here naphthalene ring and that is substituted with the OC2H5, that is as a R. NHCOR, now R we have to replace with the naphthalene ring with the OC2H5. Now one more drug, that is a prodrug for aminopenicillin. So this is what aminopenicillin, benzyl penicillin is here. Likewise just we need to change at the COH. COH is changed to the ester. As we have seen in a SAR, this COH group is changed, hydrogen of COH is changed with this moiety and become the ester. That is that gives us a prodrug, example becampicillin. So this is what benzyl penicillin as we are having, and this is the change in ester and we are getting becampicillin as a product. These are the market preparation. Now coming to the again penicillinase resistant drug, oxacillin. So again similar to that of natural penicillin, here the common structure all the side chain as NHCO. Now NHCO has here R. So R is here we have to replace by the name Oxa. Oxa indicate their oxazole. So we have to directly attach here NHCOR. After CO we have to directly attach this oxazole, and this oxazole substituted with this one benzene ring and another here is here methyl. Okay, so that is a oxazole. So if you know this structure, that will be easy to get this second molecule, this third molecule and this is the fourth molecule. So likewise you will get the next 4 molecules. So you should know the structure of oxacillin. Coming to this second, cloxacillin. The word indicate here, the name of drug indicate here oxacillin as it is, but it is substituted with the chlorine. Cl indicate the presence of chlorine. You can see here the chlorine. So this is what a chlorine is present in that rest of part. Next is here dicloxacillin. As we are knowing cloxacillin, it will be easy to get the next molecule, that is a di. Di indicate the presence of two. Diclo means two chlorine. So one, as we can see now. Next is here on the phenyl ring, which is attached to the oxazole. So likewise you are getting cloxacillin from oxacillin, and from cloxacillin you are getting the dicloxacillin. Now from dicloxacillin again you will get the flucloxacillin. So flucloxacillin is here, indicate flu means fluorine atom, Cl indicate the presence of chlorine, and oxacillin as we are knowing the structure of oxacillin. So here one chlorine is there and flu, that indicates the presence of fluorine. So here the fluorine is present. So it becomes flucloxacillin. So by that way, from one structure we are getting number of molecules. Now for what purposes we can use this penicillin? So free penicillin form is not suitable for oral and parenteral administration, so it is utilized in their either sodium salt or potassium salt. These are the different uses for the penicillin. It can be used for pneumococcal infection, streptococcal infection, streptococcal pneumonia, arthritis, meningitis, endocarditis, staphylococcal infection, meningococcal infection, gonococcal infection, syphilis, diphtheria, anthrax, clostridia infections, and for surgical procedure in patient with the valvular heart diseases. Adverse effect. The main side effect of the penicillin is hypersensitivity and the pain at the site of injection. So diarrhea, that's the one side effect. Fever, chill, body ache, flu symptom, urinating less than usual or not at all, severe skin rashes, itching or peeling, agitation, confusion, unusual thoughts or behavior, seizure, then you can see the nausea, vomiting, stomach pain, vaginal itching or discharge, heartburn, headache, and thrush. So this is all about the side effect of the penicillin. So hope you understood about the classification of antibiotic, and then we have discussed about the penicillin, their mode of action, and SAR, their uses and side effect. Now here again I want to tell you about how to draw these all structure which we have discussed here. So take a look on that video. Today here I am going to tell you about how to draw the penicillin analogues by their name. So let's see first of all the basic nucleus what it contains. The beta lactam. It is connected to the another ring, the adenosine, and at sixth position it contains the acyl amino side chain. So here we are going to change the different molecules and we are getting different molecules. So by the name we will get the structure. So let's see the first example, benzyl penicillin. So this is what a basic molecule, what you can see here. Just we have to change this one. So here benzyl indicating the CH2 and this one. So this is just we have to substitute that benzyl part here. So this is what we are getting the benzyl and rest of part is common, penicillin. Coming to the another drug, phenoxymethyl penicillin. This we are getting from the benzyl. So in the series of penicillin drug, we can get the another molecule from the previous one. So here sequence is also very important. This is what a phenoxy that you have to substitute. Okay here, and next is methyl. So methyl will be here as it is, and just you have to substitute here as this one. So like this phenoxy, methyl, penicillin. Coming to the other one, ampicillin. So again come back to the structure of penicillin. Benzyl penicillin. This is what we are having. Benzyl penicillin. Now in this case, here we have to introduce amino group. Indicating the presence of amino group. So we have to introduce amino group. So here where we are having the structure of benzyl penicillin, just one of the hydrogen is here replaced with the amino group. So it become ampicillin. Rest of part is same. Coming to the next, amoxicillin. So again it contains the amino group as it is here. Hydroxy. OX for oxy for indicating hydrox. Just remember oxy for it gives us A for hydroxy. So here at the para position we have to substitute hydroxy group and it become amoxicillin. Now similarly again come back to the structure of benzyl penicillin. Okay, we are having this. Now coming to the carboxy penicillin. Introduction of carboxy ARB, introduction of carboxy group. So here what we are having again hydrogen we have to replace with the COH. So in the case of ampicillin we are having the structure NH2. Now in the case of carboxy we have replaced NH2 of ampicillin with the carboxy group, so it becomes carboxylin. Now next to this from same series, picarcillin. So it indicates T for remember for thiazole. Okay, remember for thiazole, and next is carboxy group, and as it is our common part. So here carboxy group common part, and here we have replaced the phenyl ring with the thiazole. So it becomes picarcillin. Now coming to the other, so sequence is very important when we are doing this series of drugs. So next is methicillin. So again cilin is common part as you can see here. Now methyl they indicate presence of methoxy group. So here directly get this what we are having this R, NHCOR. So NHCO. Now directly R we are going to substitute here the phenyl ring, and adjacent carbon contain methoxy group. So it is a methicillin. Next to this naphthalene. The word will indicate Na for naphthalene, presence of naphthalene ring. So here make it one drug from the previous one. So likewise we are doing this naphthalene, and here at the adjacent one carbon, right, give sufficient ethoxy. So it becomes naphthalene. Coming to the other examples, oxazoline. So here again we are having the general series, common part, beta-lactam, dihydrothioazine, which is fused to this beta-lactam and gives us penicillin. Coming to this sixth position, this is the common part. Now here in the case of oxazoline, from one to another. We can read from their name only. Thank you. So hope you have enjoyed this session and understood all. Thank you.